students. Today we'll start part 2 of biomolecules, class 12 science chemistry. Okay, so today we'll pick up the topic protein. You already know what is protein. Proteins are the polymer of alpha amino acid. These are also known as polypeptide. Now, how the peptide bond form? You see, we have taken one amino acid here where NH2 group present and one carboxyl acid group is present. Now, if you have chosen two amino acid, there will be a condensation reaction in between them. In condensation reaction, a small molecule leaves the reactant. So, from one amino acid, this OH group and one hydrogen from this amine group will combine to form water. They will leave and will form this new bonding between carbon oxygen and nitrogen hydrogen. So this bond is called peptide bond. So in protein we get many peptide bonds since it is a polymer of amino acid and hence it is called polypeptide. Let's see some properties of amino acid. Amino acids are usually colorless crystalline solid. They are water soluble, high melting solid and behaves like salt rather than simple amines or carboxylic acid. We'll understand why they act as salt rather than showing acidic or basic character, that of amines and carboxylic acid. Next, this behavior is due to presence of both acidic and basic groups in the same molecule. They have amino acid. The term itself is saying it have two parts. One is amine part that is basic in nature, and acid part is the carboxylic acid part. So both are there in the same molecule. Now we know that proteins are the alpha amino acid. Let's understand what is that alpha, beta and gamma amino acids are actually. In this molecule you see this COH group and NH2 group, okay, both are joined to a carbon center. Now carboxyl group is more prior than that of your amine group, so this group is joined to this carbon which is alpha carbon. Now amine group, if it is connected to alpha carbon, that amino acid is called alpha amino acid. In this case you see this carbon is alpha carbon because carboxyl group is attached here, so this is your alpha carbon. Next carbon is beta carbon. Amine group is connected to beta carbon and hence it is your beta amino acid. In this case you are getting alpha, beta and gamma where amino acid is attached. On gamma carbon amine group is attached and hence it is your gamma amino acid. So in protein, what type of amino acid we get? We get this amino acid, that is alpha amino acid, where R is the side chain. R may be anything, it may be hydrogen, it may be CH2, it might have a benzene group. Depending on that we get 20 kind of alpha amino acid which basically make the proteins. Next we'll see the amino acid classification. Amino acids are classified as acidic, basic or neutral depending on the relative number of amino and carboxylic group present in the molecule. This is an example of alanine. Do remember the simplest amino acid is a glycine where the R group is hydrogen. In alanine the R group is CH3, it's a methyl group attached. This is the carbon center. This center is the carbon center. Now in this molecule you are getting one carboxylic group and one amine group. So one acid group and one basic group. The number is equal, so this one is your neutral amino acid. In aspartic acid you see you are having one NH2 group and two carboxylic group. So acid group is more than that of your base. So this one is your acidic amino acid. In this case you are getting 1, 2, 3, 4 amine group and one carboxylic group. Basic group is more, so this one is your basic amino acid. So depending on the number of amine group and carboxylic group, you can easily classify the amino acid as neutral, basic or acidic. In amino acid, we get one acidic group and one basic group. Here nitrogen contain lone pair of electron on it, right? It's a basic group. Acid have a tendency to donate a proton and base have a tendency to accept a proton, that all of us know. Now in the same molecule, what happens, the acidic group, that means carboxylic group, will release the proton and the proton will hook up with the nitrogen because it possesses the lone pair of electron. So there is a shift of proton within the molecule. As a result, what happens, carboxyl group generate a negative charge because it loses the H plus ion, and H plus ion added up in the amine group, and hence one positive charge develop over here. So we get an ionic form of the amino acid, and this form of amino acid is called zwitter ion. This part is very important. What is zwitter ion? So zwitter ion is the ionic form of the amino acid. Electrically, this one is neutral because you are having equal amount of charge, 1 plus 1 minus. It's a neutral compound, but it is dipolar in nature, okay, and it reflects the amphoteric nature of the amino acid. That means amino acid can act as an acid as well as a base, and in zwitter ion form it acts as a salt because it have a plus minus charge. It is electrically neutral. Next, let's see what are the different types of amino acid. Now amino acid, based on their function, can be classified into two types, that is essential amino acid and non-essential amino acid. Essential amino acids are the one that our body cannot synthesize. Do remember this one, our body cannot synthesize, so we have to take it from outside through our food, through our diet. These are called essential amino acid. Example, valine and leucine. Now non-essential amino acids are the one which can be synthesized within the body, so we need not to take it from outside. Our body itself can produce it, and hence it's a non-essential. Example, glycine and alanine. Next, isoelectric point of the protein or isoelectric point of the amino acid. Isoelectric point is the pH, okay, this is not temperature or something, this is the pH. This point is the pH at which dipolar ion exists as neutral ion and does not migrate to the electrode like cathode or on anode. This point is called isoelectric point. So at a particular pH, what happen, the zwitter ion or the amino acid, they stop their migration. They have plus charge, they have minus charge in equal amount. So whenever you put an electrode, they neither shift to the cathode nor to the anode, and that particular pH is called isoelectric point. So isoelectric point is the pH at which amino acid do not migrate. At isoelectric point, the solubility of the amino acid is very less. So if you want to take out the protein at this point, you can coagulate the protein easily and can take it out because they are present in the most insoluble form at this stage. Next is denaturation of protein. Now what is protein denaturation? This is a process in which a protein loses its native structure by application of some physical change. For example, if you are changing the temperature, if you are changing the pH, then protein generally loses their native state, okay, and this process is called denaturation. In native state, what happens in this denaturation, protein loses its quaternary structure, tertiary structure or secondary structure. All of this will convert it to primary structure, but in denaturation of protein, primary structure do not get affected because in primary structure you are just getting the sequence of amino acid, which is a chemical change, okay, but all this happened due to some bonding. The bond break, the bond, that's simple bond, okay, hydrogen bond, then covalent bond, this can be rearranged, but it is very difficult to change the sequence of amino acid. If you are changing the sequence of amino acid, the protein itself will change. So structure of protein change in denaturation, but the protein sequence That means amino acid sequence present in the protein do not change. So secondary, tertiary, quaternary structure get affected in protein denaturation, but not the primary structure. You have to remember this. Now, protein denaturation leads to the loss of biological activity. The biological function of the protein also changes in the denaturation process. Okay. You see, this is the native structure of protein. When you are applying heat, you are changing the pH by adding some acid, base or electrolyte salt, then they become simple. That means from complex structure they are converted into the primary structure, where the sequence are same but the structure is different from that of your native state. One example is denaturation of the egg albumin. Albumin is a protein present in egg. Now you see this liquid egg, okay? Here if you are applying heat, then what happen? The liquid part get coagulated, okay? With application of heat, it get coagulated. Sometimes the denaturation is reversible, but most of the cases these are irreversible too, and it happen by these factors: heat, acid, salt, and other mechanical process. Now, what is the role of protein? Protein act as enzyme and also as transport agent. In hemoglobin, the globin protein, the globular protein is there, which carries the oxygen from your lungs to the cell, then cell to the lungs, like that. So it acts as a transport agent as well. They are structural material of nails, hair, cell, etc. Okay, these are the basic the protein. The term protein itself came from proteose. That means the prime uh, prime functional group. Okay, prime importance. So these are something that is the that make the basic structure of the body. It is present in the cell. The cell accumulate to make the tissue, then the tissues are forming organ, and the organs are making actually your whole body, like that. So these are the structural material. Next, antibodies formed in the body are globular protein. They are metabolic regulator like insulin, etc. So these are some roles of proteins. Next we'll come to the enzyme. Now what is the enzyme? Enzymes are the globular protein, and enzymes are biological catalysts. These are not chemical catalysts, but these are the catalysts present in the body, so they are known as the biological catalyst. Some important enzymes are lactase, that convert lactose to glucose or galactose. Okay, lactase, where it is present? It is present in the milk. So lactase enzyme can convert the lactose sugar present in the milk into galactose and glucose. Lactose is a disaccharide, if you remember. Then invertase convert a sucrose to glucose and fructose. Maltase convert maltose to 2-glucose unit. These three already we have studied. Then emulsion convert cellulose into glucose. Urease convert urea to carbon dioxide and water, and many more are there. Okay, so this is some important enzyme. Mark one thing, the name of the enzyme always end with ASE. Okay, S, lactase, invertase, like that. Next, enzyme mechanism. How the enzyme work? We know that enzyme is a biocatalyst. This is highly specific in nature. Okay, and in any reaction, the catalyst always used in a small amount. In a very small amount, they work. Now, since they are very much specific in nature, and since they act as a catalyst means what? In a reaction they will take part, but there will be no change. They will remain intact throughout the reaction. So see the mechanism. We have chosen one enzyme. Okay, this is your enzyme, and here substrate. That means the reactant are coming. See the shape, see the surface of this enzyme. It is very much specific in nature. That means specific substrate can only get added up over here. So substrate will come here, they will combine to form a intermediate state, that is ES. Now after getting attached over here, this one will break down. This one will convert into EP, intermediate state. Afterwards they will separate, and you are getting these two product, and you see the enzyme is intact. So it is acting like a lock and key. It is like a key. It is coming to the lock, then the in the key some change occurs and they are going back. The lock remain intact. Okay, so this mechanism of enzyme is called lock and key mechanism. So these are some few highlights about your protein. Rest of the things are known to you. This structure of protein is very much important. All of you had make the note, but do remember there you have to add the diagram in the note. Please add the diagram for each structural proteins. Okay, so I'll finish today. That much for today. Thank you.